Hello everyone, my name is Dr. Robson Leite. I'm a PhD in Marine Biology under the guidance of Drs. Mara Kiefer and Daniel Lima from Universidade Federal Fluminense. Today I'm here to present part of my doctoral thesis entitled Structure and Temporal Dynamics of Brachyura and Crab Assemblages in Mangrove Stands of a Coastal Lagoon in southeastern Brazil. Mangroves are unique tropical and subtropical ecosystems. They are transitional habitats between land and sea, associated with rivers, estuaries, and bays. These complex ecosystems are constantly exposed to tidal fluid and to a set of dynamic environmental variables. Mangroves are highly productive and export organic matter to the estuary. Therefore, they represent favorable habitats for feeding and reproduction for a variety of species. That's why they are known as natural nurseries. Associated with mangroves, Brachyura crabs have great ecological importance due to their high abundance and functional roles. Their bioturbating activity and detritivorous feeding contributes to the cycling of organic matter. They are highly diverse and are dominant in mangroves. Some species have economic importance as they serve as food. There is a paucity of knowledge when comparing ecological processes between natural and planted mangroves. This information allows evaluation of the impact and efficiency of these interventions in relation to the facilitation of natural colonization. Therefore, the main objective of this study was to describe and compare the structure and temporal dynamics of Brachyura between two lagoon mangrove stems of distinct origins and their interrelationships among environmental factors. At the coast of Niterói, Rio de Janeiro, Brazil, a naturally colonized mangrove stem emerges in the Itaipu Lagoon after the opening of an estuarine channel in 1977. In 2003, his offramang plantations were carried out at the opposite margin in a non-vegetated soil. Therefore, two distinct mangrove stems emerges as a mosaic of structural development. As a new ecosystem, little is known about their crab assemblage. Given the ecological significance of mangroves and crabs, these stands provide an excellent model to assess the responses of the group to the different habitat features. Sampling took place monthly over a year. Because of the variety of crab niches and microhabitats, direct and indirect methods were carried out, being crabs and burrows. Abundance of individuals were compared between stands, and along with environmental factors, were explored in multivariate statistics. Density of burrows were compared between stands. Salinity and productivity were higher than natural stand. The richness was 9 species within 6 families. Ocypodidae and Sesamidae were the most representative. The species sampled are shown. Species abundance with its different direct sampling methods are shown, with its total abundance being the sum of them. Minuca burgesi and Erythro mimosa were exclusive to the natural stand. Minuca burgesi abundance was not quantified due to taxonomic issues. Minuca hapax was the most common species. According to Anocina and Simper results, Minuca hapax and Euelis granulata contributed more to the dissimilarity between stands, which was 27%. Density of Minuca hapax was higher in the natural stand. The PCA model is represented with 4 principal components, and crabs and environmental factors were better associated at the natural stand in the rainy season. Low richness is expected in the neotropics, as opposed to paleotropics. The most representative families are consistent with the literature. Richness is correlated with area size and developmental stage. Smaller and younger mangroves, dominated by Laguncularia racemosa, tend to have lower richness due to their lower structural complexity. This is the case of the planted stand. However, both stands of the lagoon tend to be less complex when their richness is compared to some mangroves of similar size. Fiddler crabs are dominant in most mangroves, and Minuca hapax is usually most abundant among them. Species were more abundant at the natural stand. The interrelationships in this environment are justified by size, age, forest structure, productivity, and tidal input. Hence, it's more structurally complex, promoting niche variability. It's a more saline and productive environment, which corroborates the presence of Lucids in Euryalline species. The litter production was similar among stands, though. Despite their differences, fauna difference was subtle. Also, the close distance between stands could promote larval dispersal and thus enhance similarity. In conclusion, the natural stands favor the greater distribution and abundance of crabs, since the set of its characteristics justify its greater structural complexity. Despite the stands' differences, the crab composition was similar. One reasonable explanation is the proximity of the stands in the small lagoon favoring dispersion. In addition, his offer among planting was successful in favoring the colonization of crabs and the development of planted stand, suggesting that they tend to fulfill similar ecological functions. That's all. Thank you for listening and for the opportunity to share my work. See you all soon.